Hey everybody, we're back today and we're going to talk about Chapter 9 in the topic of genetics. So let's go ahead and jump right in here. So genetics is a really broad field and there's a lot of things to talk about. So we're going to condense the topic down quite a bit and we're going to look at it from a perspective of these four things here: transmission of traits, expression, variation, structure and function, and then change in genetic material. So first of all, genetics kind of traditionally is viewed as the study of inheritance or the heredity of living things. So in other words, heredity is in reference to the transmission of your genetics from parent to offspring. The truth is, it's a lot more involved than this. This is the definition out of the text. This is kind of a traditional definition. And really, it has so much more to it than that, and so that's what this list down here is talking about. It does talk about transmission of traits, but it goes on to talk about what we call expression. I'll mention what that is. We won't talk as much about variation of traits. That's more of a principles of biology, but we will get into the structure and the function, and we will talk about change in genetic material, which really is talking about mutation for the most part. We'll also have a few other examples in relation to bacteria, since they do some things that other organisms really don't do. So that's kind of the overall set of topics here. So in this next part, I want to mention the fact that genetics is something that takes place in multiple levels. So traditionally, it's always been done in an organismal sense. And when you're looking at, for example, how traits are passed from parent to offspring, you would study that for the most part, especially traditionally anyway, just by looking at the offspring and comparing them to the parents or vice versa. So if you took general biology, you may be familiar with Mendelian genetics, and Gregor Mendel, the father of this particular area, was responsible for looking at things like dominant and recessive traits and how offspring would look similar or different from parents based on some of these traits. So anyway, that's a very classical view. As technology increased and progressed, it gave scientists and researchers a better way to actually analyze this. And really, around the same time, they were simply discovering what it was that actually led to the traits. So really early on, they didn't actually know why they had those traits, but they could study and actually watch some of the patterns that they would have. So they were able to actually figure out a lot of it before they even really knew what it was, just based on certain patterns. But when you look at cellular and chromosomal genetics, you're really looking at specific details within the cells, and you're looking at a very, very close-up look, so to speak, of what's happening. And most of this research here took place in the early 1900s to the mid, even late 1900s. Really around the 1970s and going forward, we saw a much more rapid shift towards molecular genetics, which is really where the field largely revolves today. I mean, ultimately, you're now really studying all levels, is the truth, but molecular genetics is studying DNA at a very specific, highly detailed level. So you're getting down to the very, very specific sequences that make all the difference. And so really, when you have that ability of control, you're going to use it. And they can go back and work upwards to higher levels. But really, this is where the field takes place today. So next here, we're going to talk about some of the terminology, just real quick. And so first of all, one term you want to be familiar with is the term genome. So the term genome is essentially the sum total of an organism's genetic material. So it's basically all the DNA that makes an individual or an organism what they are. So you could say the human genome, and that would discuss all the genes of humans. Genomics is the study of these large genomes. Okay, now a genome, and really let's go back and just talk about a fundamental thing here, because I kind of skipped over this. DNA, we're talking about genetics, and genetics really is another way to think about it is, and this is not really the definition, but you can think about it as the study of, sorry about that, the study of DNA. Just a second here. Yeah, put that in air quotes. Just a way to think about it. Now, we can think of DNA then. You think of DNA as a biological set of instructions. Another way to think about it is, this is a reference you hear a lot, like a genetic cookbook. Got lots of recipes in it. So another way to think about it is like a biological blueprint. So it's got a lot of information that the cell is going to use to activate different parts of the cell, and ultimately for the cell to become what it is. So essentially the DNA is a set of instructions that basically guides the entire creation of the cell, as well as all of the characteristics of the cell. Okay, and that exactly how that works is really the whole, the whole sum of genetics. So it's not something that's easily understood. So we're going to start with some concepts, and then we'll try and work into actually how that works. So anyway, to start, we want to just understand DNA is like a set of instructions. And another way, part of an analogy I use, is you can think of DNA as a book. Okay, a book of information. So if DNA is a book, then essentially our genome is the entire book. Okay, that's the sum total. The next level that we can look at DNA at is the chromosome level. And we can think of chromosomes really as chapters of the book. So a chromosome contains segments of the overall genome. Each organism has a different number of chromosomes, and this is a number that's referred to as an organism's chromosome number. Every organism has a unique, well, I shouldn't say every organism has a unique chromosome number, but every organism has their own chromosome number, and they're different for every organism. So if we just take a quick look here and type in chromosome number, we'll see that we get, if my internet was working, we would see that. Sorry, try that again. We get a lot of different numbers for different organisms. So here's a good example. And we can see that humans have 46, fruit flies have 8, rats have 42. A random example here, the fen duck has 80. Never saw that one before. Tobacco, 48, etc. Another interesting thing to point out here is that the organism's complexity is not at all related to its chromosome number. So in other words, more is not better. For example, tobacco actually has 2 more chromosomes than a human being. And I think tobacco is safe to say a little bit simpler than a human being. So interestingly, that chromosome numbers are not equal to the complexity. More is not better. But anyway, every organism has their own number. So anyway, every organism, like every book, has like a different number of chapters, which kind of fits that analogy. The last part that we're going to look at are what are referred to as the genes. And I apologize, I should have put this slide up here a second ago. This is talking about chromosomes as being discrete cellular structures that have neatly packaged DNA. And that's something, I should probably mention that real quickly here. Let me just point that out. The way a DNA molecule, or the way a chromosome is structured is a little bit different. So, for example, eukaryotic chromosomes are typically linear. They're relatively straight, whereas prokaryotic chromosomes are actually circular. And what that actually means is that they're linear, but they connect end to end like that. So they essentially double back on themselves and connect, and that makes them circular. Whereas eukaryotic chromosomes don't ever do that. Being linear actually allows eukaryotic chromosomes to form tighter bundles, and typically what happens is these big strands will actually coil and wrap around themselves into really dense bundles, and that actually can create a chromosome that's in a more packaged and tight bundle. So here's a good figure showing that how DNA starts out kind of a linear form and is actually wrapped around these proteins and then folds into a tighter and tighter strand until ultimately that folds into what we see as a chromosome here. So eukaryotic chromosomes fold into these highly complex condensed bundles. And why that's somewhat significant is because, amazingly, complex organisms like humans, we have about 6.5 feet of DNA per cell, which is insane, because if you take an individual cell, if I put this dot right here on the computer screen, right there, you could probably fit 1,000 cells on that dot, human cells, something like that. Maybe I'm a little bit off, but probably 1,000 cells could fit on that little dot right there, and each one of them would have 6.5 feet of DNA in it, which is kind of mind-boggling. The only way that actually works is wrapping it really, really, really tightly and bundling it very close. So there's a huge difference in how DNA is organized. Eukaryotic, being linear, are really, really big and tightly bundled. Prokaryotic chromosomes are typically much smaller, so they don't have nearly as much DNA. They're very simple organisms, so they don't have hardly any amount of DNA as a complex eukaryote. So they typically have less organization in a very different structure, being a circle. This figure from the text is showing the structure here in some of these different organisms. It actually talks about viruses a little bit. I kind of skipped over that just to kind of condense this down here. But interestingly, viruses have DNA and RNA that is similar to, or really almost identical actually, to other organisms like bacteria and eukaryotes. And so there's a lot to talk about there, but I'm going to not get into that. So the next part here is going to look at the next level of organization, which is the gene. So going back here, we looked at organisms, and I described the genome as the sum total, chromosomes as being like a chapter, a certain section of the overall whole. And the last part here is what we refer to as a gene, or genes. And following this analogy here, you can kind of think of genes as being like a paragraph or a sentence of the book. So they're a specific segment that has specific information for the cell. And next we're going to talk about kind of what's happening and what this information does. But when we think about the organization, we think of genetics as the study of DNA. DNA has all this information, and it's organized at different levels. And really, when we get down to the gene level, to connect back here, this is really what molecular genetics is looking at. Either looking at individual genes, sometimes small clusters of genes, or even certain regions of a gene. And there's a lot to be done with just looking at a small piece of a gene. So that's some of the different levels there. Now, this next part here goes on to describe what is a gene. And this is actually kind of an interesting question, because early on it was described differently with a lesser understanding, and today with more information it's described a little bit differently. But both ways actually work, and that's why I talk about both of them here, and the textbook mentions both. So, for example, the classical view describes a gene as a fundamental unit of heredity responsible for a given trait. And so it's essentially a part of your DNA that gives you some sort of feature in your body, or a feature of a cell, or something like that. And traditionally, it's kind of something that you can either see or measure, or something that is somehow apparent that it's there. So a trait, something that you can you're aware of to a certain degree. And really, in a classical standpoint, that was the only way they could study it, because you didn't know what you couldn't see or somehow measure. Today, though, there's so much technology and there's so many different ways of analyzing. They now can look at information in the cell without really even having to see any characteristic of the cell. They're just looking at the DNA and ignoring the rest of the cell, or the organism. So from a molecular biochemical standpoint, what they now describe it on is really just as a site on the chromosome that provides information for a cell function. A lot of times that information ultimately creates a trait. So as an example, you can have a gene that codes for a protein. And the protein creates a reaction that creates a pigment. A pigment is a molecule. A molecule that gives color, and that's not the definition. That's just a way to describe it. So a pigment is a molecule that gives color. So you can have a gene that creates a protein that creates a pigment, and the pigment then provides a color that you can actually see with the naked eye. So that's an example of a trait that you would actually see, but at the same time, you could have a gene that codes for a protein that, for example, codes for a cell membrane transport. It would be a cell membrane transport protein, so I don't want that to be confusing, but you can actually have proteins that make other proteins. That's a lot of times the way it works. But you essentially have a gene that creates an enzyme, really is what this would be, that ultimately creates a cell membrane transport protein. So you have, in this case, a trait that you would never see. You would only know that this is there if you actually were able to see a cell or measure it somehow indirectly. So it's still a trait, to be honest. It's just not the same way that you would think of a trait from a classical standpoint. So from these two views, you kind of get a different look at it. And the last part here goes back to the central dogma of biology, which we'll talk about here in just a second, and that is a certain segment of DNA that contains information to make a protein or RNA. And in fact, you know what? Let's just go ahead and skip to that real quick. So we were going to look at that here. Central dogma of biology. So let's go ahead and just put that up here. And I know we've talked about that, and hopefully you remember. And if not, this is a good time to review. But if you do, then you're ahead of the game. But if you recall, the central dogma of biology states that DNA codes for RNA, and RNA codes for protein. And then, so to clarify this is the central dogma on its own, and then I like to then go on to say that protein does the quote-unquote work of the cell. Protein is also responsible for many of the structures of the cell. Lots of other things. Those are just kind of generic ways to describe what's happening. So that's the central dogma. So really going back and looking at this generally speaking here, this is, by the way, out of the textbook, and the textbook just describes this as generally speaking. So it talks about a classical view, and then it talks about a biochemical molecular view, and then it just says, and generally speaking, it's a segment of the DNA that contains the information or the code to make RNA or protein. And so that goes back to the central dogma. And then we can see that DNA ultimately guides the formation of RNA and protein. So it does this really by the way it holds information. So there's information here that allows for these two processes to take place. And we're going to talk about how that works. It's difficult to understand, but we're going to talk about it, and we're going to try to understand how that works. Let's go ahead and look at the second part of this here, and then I'll go back to the slide I skipped. This next part here, how has the central dogma changed over the last decade with new information? Well, what they now know is that, in fact, RNA is now, is not now suddenly, but is now recognized to do a lot of the work and sometimes provide structure, but really more, it does a lot of the work that proteins are now recognized not to do. And really, it's not that there's less work going on with protein, is that there's a lot more happening with RNA. So RNA is now recognized to do a lot more in the cell than what it was thought. And previously, it was thought that RNA is more or less just to go between with a certain. There are some recognized examples that have been around for a long time where they knew it didn't necessarily always do that, but for the most part, it was kind of an intermediate between the ultimate goal of protein. But now they know that there's actually a lot going on with RNA, and it's a lot more complex than was once thought. So to give you an example of some of the work in this particular case, gene regulation. Genes, you can think of these as on-off switches for genes. So an RNA molecule, for example, can act to turn on or off a gene, which can then regulate when a protein is produced. So this gives actually some feedback that the cell can use to determine when to make a protein. So an RNA molecule can be produced first, and then that goes on to regulate when proteins are produced later. That's just one example, but that's one way the RNA does work. A big example, because it's one of the primary things that they're recognizing the RNA does. So anyway, that's not something I'm going to ask you about a lot of detail of, so don't worry about that too much. Just know that RNA does more of the work than previously thought, okay? And that's really all I want you to know about this part here. How has this dogma changed with new information? RNA does a lot more work than thought. Gene regulation is an example. Let's go back to this last slide here that I skipped, and then we'll take a break, and or rather have you do some review questions. I'm going to take a break, and then you guys can do the review questions. So gene categories. When we look at genes, as I just said, most of them ultimately produce protein. Okay, so to clarify, this is still mostly what's happening, but it's now recognized that lots of RNA is happening too. The percentage, I'm not honestly sure. There's at least 20,000 proteins produced in a cell, in a human cell. I don't think there's anywhere near that many RNA, so I don't think it's like 50-50 or anything like that. So protein is still doing most of the work. And why I'm saying that is because a large percentage of our genes, or a big portion anyway, are what are called structural genes. It's actually not a large percent of our DNA, but a lot of the active DNA are considered structural genes. So these are genes that code for protein, and these are just kind of the traditional view with the central dogma of biology. These next ones here are really kind of exceptions to that central dogma, and these are different layers of complexity in this whole discussion. So the central dogma doesn't describe everything. RNA playing a bigger role, is an example of that. So RNA genes is another example, and this is another way that RNA comes back into the picture. So here we have a gene that only codes for RNA, and the gene is not intended to ultimately code for protein. So RNA is really kind of the endpoint here, and we create molecules of RNA that then go on to carry out functions in the cell. And I know that doesn't make a whole lot of sense. The problem is there's a lot more you need to know before that makes any more sense. So unless you already have some genetic background, that probably won't make any sense. I do apologize. But as an example, we're going to talk about transfer, little tRNA, little t, capital RNA. Transfer RNA is an example, and this molecule here is exclusively RNA, and it's going to play a role in a process called, I don't know, I need air quotes on that, a process called transcription. Okay, we'll talk about that here in a minute. So when we look back, if you look back at this will make more sense later. But that's an example of where you have an RNA gene that doesn't produce protein. The end goal here is tRNA, transfer RNA, and that's something that's going to do some work, quote unquote, later on. So that's an example of an RNA gene. The last item, the regulatory genes, and many of these are also RNA, as I mentioned, the role here in work in gene regulation. So they now recognize that, in fact, there's a lot of these genes. Again, I don't know the numbers, but there are, in fact, a large number of regulatory genes that control protein production. And to kind of give you a perspective on the difference here between these genes, let's talk about structural genes versus regulatory genes, okay? And these are kind of the two primary types, really. RNA genes here are kind of a small percentage. So really structural and regulatory are the two big ones. You can think of it in the terms of a structural gene is information for how to make a tool. Okay, how to make also how to make building material. So in this case, we're talking about cellular, of course. But if we use an example of like construction, like a home construction, you can think of the structural genes as providing the tools that you would need as well as the building material for how to make a house. So you need boards, you need cement, you need hammers, you need nails, you need screwdrivers, etc., etc., etc. Lots of tools, right? Lots of tools to build a house, lots of building material to build the house. Structural genes essentially give the cell that material. It provides them the information for how to get those things. Regulatory genes tell the cell how to use the tools and materials, the building materials. So having the materials and the tools doesn't do anything for you if you don't know how to use them. And this is where things like hammers and nails and boards and screws, etc., are going to be much better off in the hands of a craftsman than they are in the hands of, say, a biologist. So the difference here is having the tools versus knowing how to use them. And it turns out, interestingly, that this apparently is one of the biggest areas where the DNA of different organisms differs. And this is actually kind of interesting. So I'll give you a couple examples here. In fact, let's just look it up. There is a surprisingly large amount of DNA in common with different organisms. So let's see here. Human DNA in common. Yeah, how about that one? Banana. Now, I'm not vetting my sources here, so if I'm off, then shoot me. But this is pretty consistent with what I would have guessed it to be, so I'm assuming this is somewhat accurate. That the value is right around 50 to 40%, if somewhere in that neighborhood. So you're talking almost half, if not at least half. So maybe this, here's one. Yeah, that's, anyway, if I'm wrong again, it's something like 40 to 50%. And the reason why this is the case is because, amazingly, at the cellular level, a lot of organisms produce the same stuff. If you think back to chapter 5 on cells, especially eukaryotic cells, because to clarify, bananas and humans were both eukaryotes, so we both have complex cells. There's a lot going on at the cell level that accounts for a huge amount of the DNA in an organism. So a huge amount of the DNA is designed to just code for basic cells and cell functions. And a lot of that's structural genes. And some of those are also regulatory. But where you start to get a lot of difference is when you start looking at how the DNA is used. So I'll give you another example here. This one I know I don't have to look up. Chimpanzees, chimps, and humans, we have about 99% of our DNA in common. And in fact, let's just make sure that's correct, because it's greater than 98%. Okay, maybe I'm 1% off.
On behalf of the team at Creation Ministries International, thank you for your first-time visit.
That's enough. Okay, well, greater than 98%. So, anyway, it's a really large percent. I was always told 99%. So 98, 99%, something like that. It's good to look these things up. Maybe I'm a little bit off here. 98%. So we have a huge amount of DNA in common, okay? And we're very, very similar, even though we look very, very different. So how is this possible? Well, we basically make all the same stuff. Our body has all the same types of skin, hair, teeth, bone, nerves, really, really similar. From a building material standpoint, we're almost the exact same thing. It's like two houses that are built out of the exact same materials, but you have two different builders that make them. They're going to look different, but they're more or less the exact same thing. So what makes all the difference is how the tools and the building material is used. So for example, if you think about chimps have hair all over their body. So in humans have little hair on the body. Obviously there's some exceptions to that, but most people are relatively hairless in comparison to chimps. But we both make hair, and the hair on both of our bodies is very, very similar. So the ability to make hair and make it out of something both come from structural genes. The difference in regulatory genes would dictate how that hair is made, where in the body it's made, and how much of it's made. So whether we're making it on our chest, whether we're making it on our buttocks, whether we're making it on our face, whether we're making it on our back, etc., all these different areas, is determined by regulatory genes. When it's made, where it's made, how much it's made. Structural genes determine that you can make hair, and then regulatory determine where and how much. So that's how, you can apply that example to almost everything in the chimps and human bodies. So I'm using hair as an obvious example, but this is going to apply to almost everything. And that's why we have so much DNA in common. The 2% is mostly going to be in regulatory genes. How the materials are used. And when it comes to chimps and humans, some of the differences come, for example, in opposable thumbs, prefrontal cortex, and foot structure, things like that. So there's some key differences that make a really big difference. But amazingly, how much DNA is common. And one more example, something like 70 or 80% with fruit flies. Let's see here. Oh, 61%. I guess I was off there. 60%. Okay, I was off. My bad. 60%. Okay, 60%. So I was exaggerating a little bit there. 60% in common with the fruit fly. It's still a huge amount, really, when you think about it. And again, it goes back to structural genes and regulatory genes. Okay, so apparently I need to brush up on my numbers here. All right, let's go ahead and, you know what? Let me. I'm going to pause it real quick. I'm going to bring the review questions up because I forgot to get those, and then I'll let you look at them. Okay, so here is a look at the review questions from part 1. Go ahead and pause this, write those down, and we'll go ahead and continue on to the next part here in just a second.